Mitosis begins with early prophase, in which the formed chromosomes begin to condense for an easier anaphase. Simultaneously, the nuclear envelope begins to break down and releases the centrioles, which move to the opposite poles of the cell. Mitotic spindle grows the microtubules to begin to capture the kinetochores of the cell. The microtubules bind the chromosomes at the kinetochore and begin to arrange them for metaphase. The extending microtubules form a structure called the aster. Step 2 of mitosis is metaphase. In metaphase, the spindle has captured all the chromosomes and has lined them up along the equator, known as the metaphase plate. Prepping for anaphase at this point, the two kinetochores of each chromosome connected to the microtubules on the opposite poles of the cell. The cell will then undergo the spindle checkpoint, which will help ensure that the sister chromatids will split evenly between the daughter cells. If not, then anaphase will not occur until the issue is resolved. The bond between the sister chromatids are broken, which will allow the sister chromatids to separate from each other and are pulled towards opposite ends of the cell. The microtubules not attached to the chromosomes will elongate and push apart, elongating the cell. Additionally, motor proteins will carry the chromosomes to the cell poles. Cytokinesis begins in anaphase as well, starting to form the cleavage furrow in which a ring of protein filaments will form around the equator just beneath the plasma membrane and will begin to separate the daughter cells. The fourth and final step of mitosis is telophase. In telophase, cell division is nearly complete as it starts to reestablish the nuclear membrane around the chromosomes. The chromosomes themselves will decondense and return to their string-like form, and the mitotic spindle will be broken down into its building blocks. As the two cells get more defined, cytokinesis will finish cinching the cells and separate them into two identical daughter cells. In plants, this will be done by a distinct cell wall forming between the two poles. In meiosis' first step, prophase 1, chromatin unravels and condenses itself into chromosomes. This chromatin is what contains the DNA and proteins within the cell. Next, the centrioles within the cell move to opposite ends. While this happens, the spindle fibers appear and connect throughout the cell. Then, the homologous chromosomes pair up with each other and form a tetrad. These chromosomes coming together is the process of synapsis. Sometimes during synapsis, the homologous chromosomes overlap with each other and can break off and swap identical sized segments of DNA strands. When this happens, they cross over with each other and they create more genetic variation. Next, the nuclear envelope and the nucleus both disappear, letting the chromosomes arrange themselves in the desired pattern. Step 2 in meiosis is metaphase 1. To start metaphase 1, the homologous pairs of chromosomes move to the center of the cell and split into a long arm structure and a small arm structure. The chromosomes split at the centromeres, and they line up on the equator of the cell by these centromeres. The cell is a round bubble sort of structure with an equator where it is at its largest point and is in the middle of the cell. The spindle fibers that are already in the cell attach to the exposed sides of the centromeres on a point called the kinetochore. The spindle fibers arrange themselves in such a pattern that the microtubules from one pole attach themselves to the kinetochore of one chromosome of each tetrad, whilst the other pole's microtubules attach themselves to the other kinetochore. Step 3 in meiosis is anaphase 1. To start the process of anaphase 1, the homologous pairs separate, but the sister chromatids remain attached to each other. The chromosomes then go through the process of segregation, where the chromosomes are moved to opposite poles from each other. Each pole should have 23 replicated chromosomes that will be used to make up the new cell's nucleus. Again, throughout this entire pushing and pulling process, the sister chromatids remain intact. With the chromosomes being pulled into their respective poles of the cell, the cleavage furrow becomes more distinct and more prominent as the one cell begins to split into its two respective cells. The protein filaments around the equator help this process. The cleavage furrow is at the beginning of the cytoplasm. The chromosomes finish moving to their opposite poles of the cell. When they finally reach their destination at their respective poles of the cell, the chromosomes stay in the form of sister chromatids. They do not uncoil and form chromatin. Although it isn't common, the nuclear envelope can occasionally reappear, but like I said, this is very rare. Finally, the cytokinesis process completes itself when the spindle fibers tighten and the protein filaments create the cleavage furrow in the middle of the cell, right along the line that will be where the cell separates into two separate cells. Meiosis also has prophase 2. Prophase 2 is extremely similar to prophase 1 in the sense that chromatin unravels and condenses itself into chromosomes, and the centrioles within the cell move to opposite ends. Also, the spindle fibers appear and connect throughout the cell. Then, the homologous chromosomes pair up with each other and form a tetrad. However, during prophase 2, there is no genetic recombination. In prophase 2, there is no crossing over between the DNA of the chromosomes. Meiosis also has metaphase 2. Metaphase 2 is very similar to metaphase 1. There is again an equator line where the chromosomes want to line up. However, instead of homologous chromosomes pairing up at the metaphase plate, single chromosomes line up at the metaphase plate. Then, the spindle fibers that are already in the cell attach to the exposed sides on the centromeres and follow the same process as metaphase 1. Once attached, they begin to contract. The seventh step in meiosis is anaphase 2. In anaphase 1, the homologous chromosomes are separated and the sister chromatids remain intact with each other. However, in anaphase 2, sister chromatids are separated. This means that an entire chromatid is taken to either pole of the cell. One side will have the duplicated chromatid, and the other side will have the original or the unduplicated chromatid. After that difference, anaphase 1 and anaphase 2 are pretty much the same. The sister chromatids are pulled to their opposite poles by the spindle fibers, creating the basis for 4 separate cells instead of just 2, while the cleavage furrow in the middle of the cell becomes more prominent and the single cell elongates itself slightly, pulling at the cleavage furrow. Finally, there is telophase 2. During telophase 2, the chromosomes are in their 4 separate groups, where they will uncoil in order to finish creating the 4 different nuclei. The nuclear envelope then assembles and forms around the chromosomes, making the nuclei of the cell complete and ready to function. Whilst that is happening, the spindle fibers disappear as they are not needed anymore, and the final step of cytokinesis happens, where the 2 cleavage furrows tighten to the point of separation due to the protein filaments tightening, creating the 4 separate cells.